Okay, a couple of announcements before we start. Your reading quiz, I told you yesterday, will be 7.1 through 7.3. That takes us through the Krebs cycle and cellular respiration. Your pre-lab quiz will not be a quiz like it's been in the past, but it's going to be your procedure that you're supposed to do. If you've looked at your lab, you're supposed to write up a procedure for what you're going to do in your particular experiment that you're doing next week. And they're due on staggered days. Monday's lab is due at the beginning of class on Monday, Tuesday for Tuesday and Wednesday, Thursday for Thursday. If you read the instructions, it asks you to write this up as a materials and methods section of a paper. We're not going to hold you to that, but there is a table in there that does need to be filled out, which basically says, for these particular experiments, this is what's going to be in tube 1, this is what's going to be in tube 2, this is what's going to be in tube 3. You're going to have that information to get yourself organized to run all of your trials and your different variables for your particular experiments. I don't know if you want to elaborate.
Yeah, it's very important that you and your lab partners sit down and talk about this ahead of time, because you've all selected a particular variable, pH, temperature, enzyme concentration, substrate, glycol concentration, whatever. That you sit down and think about this ahead of time. How are you going to vary the variable that you selected? Now, pH is somewhat selected for you. We will provide you with pH solutions from 3 to 9. Temperature, we will have a particular set of temperatures available to you. Glycol concentration, how are you going to vary it? The question is, how does varying that particular parameter affect the efficiency of the enzymatic reaction? So it's really important for you and your lab partner to sit down and say, okay, if I'm going to double the glycol concentration, so then what goes into 1, what goes into 2, what goes into 3? How much of each of the different parts? Remember, if you increase the volume of one thing, you have to decrease the amount of water. What we want you to do is sit down and think about that ahead of time, so that when it comes time to actually doing the experiments, you know exactly what you're going to do. And to help you do that, there's a table on page 6 that you can fill out that allows you to very clearly and precisely write out, okay, what's going in tube 1 for this run, what's going in tube 2, 3, and so on. This is what I want to see before you can start next week. I want to see that you and your lab partner have thought about this and have a clue as to what it is you're going to try to do. All right? Because you've got to do any one particular variable, any one pH, any one temperature, you've got to do them in triplicate. All right? So over the course of the hour and 15 minutes, you might run as many as 15, 20, 25 experiments if you're prepared. Right? Okay.
And it's important to remember that the volume of those tubes always has to be the same as you go through your various trials. And so the blank, tube 1, tube 2, and tube 3, what is it, 10 mils, I believe, that's in the blank?
5 and 5.
And then 5 in tube 1 and 5 in tube 2. So if you change glycol concentration and increase it, you're going to have to decrease your water so that the volume doesn't change from that 5 milliliters. Also remember that if you change something in one of the reaction tubes and it's in the blank, you have to change it in the blank as well. Okay? So if there's is it glycol that's in the blank, I believe?
Blank doesn't have
Peroxide, right. So if you're changing glycol, your blanks are going to change too. If you're changing peroxide, the blank won't change because there's no peroxide in the blank. Okay? So be aware of that. So any other questions? Okay, my goal is to finish cell respiration, the stuff you're going to have for the exam, by Tuesday. To do that, I'm going to have to use the Monday class period. So Monday at the beginning of last class, I'll say just a couple of words about the fermentation lab that you completed. We'll turn those back to you. There's a couple of things you need to be aware of with the lab quiz coming up. We'll then have a regular class period on Monday until the end when you take the quiz. But if things go the way I think they will, I'll be able to finish up new material for the exam on Friday. On Tuesday, that'll give you a couple days break from new material, and it'll also give us the opportunity to have some time on Thursday to review. Okay? So the goal is to finish respiration by Tuesday. Okay? All right. So we were in the process at the end of last class period of looking at kind of the big picture of cellular respiration. Remember what the goal of cellular respiration is. It's to take this energy-rich molecule, glucose, which has a bunch of hydrogens attached to it. And hydrogens represent electrons, and electrons represent potential energy. Okay? And to strip those electrons off by successively degrading glucose from one compound to another until we completely oxidize it into carbon dioxide and water. So at the end of the process, glucose is turned into CO2. All the hydrogen has been removed. Where do the hydrogens go? Well, they go first to coenzymes, but eventually the coenzymes have to get rid of the hydrogens. They'll do that by transferring them to oxygen to make water. Along the way, meaningful amounts of energy are going to be released by two processes. Substrate level phosphorylation, which is going to occur in glycolysis and the citric acid cycle. And then this very important process of oxidative phosphorylation. This is where we get the real energy payoff. Oxidative phosphorylation is associated with the electron transport system. Both the citric acid cycle and the electron transport system occur within the mitochondria. Glycolysis is going to be happening in the cytoplasm. So remember, I'm going to be using this room as kind of a model for a mitochondria. So to make sure we all understand, if you walk out that door, you are in the cytoplasm. These walls represent the outer membrane of the mitochondria. This row of chairs, which is the outside row of chairs, which goes all the way around the room, represent that inner membrane. So I am standing in the space between the outer membrane and the inner membrane. I'm in the inner membrane space. You all, most of you, are sitting in the matrix of the mitochondria. Okay? So what's going to happen is as glucose is oxidized during both glycolysis and the citric acid cycle, those hydrogens are going to be split off, but they're going to be grabbed by a coenzyme, NAD, and in the case of the citric acid cycle, FAD. And that's going to reduce those coenzymes. Okay? But remember, that's going to then deplete the oxidized forms of those coenzymes. They're all going to become reduced. And if that happens, cell respiration stops. So we need some way to convert those reduced coenzymes back to the oxidized form. That's going to happen during electron transport. And as those coenzymes are being recycled from the reduced state to the oxidized state again, as they give up their hydrogens, a hydrogen ion gradient is going to be formed. And that's going to result in protons being pumped out here into this compartment. From in here. So now I'm going to have this membrane that's going to separate this compartment, the inner membrane space, where a lot of protons are accumulating, from the matrix where there aren't many protons. That's that water behind the dam situation now. And that represents potential energy to make ATP. So what am I going to need? I'm going to need a channel protein then that allows these protons to do what they want to do naturally, and that is to go from high concentration to low concentration. And as they flow through that channel protein, ATP is going to be made by oxidative phosphorylation. But then those protons are going to have to be pumped back out here again to keep this process going. What drives that pumping? This electron transport chain, which is recycling the coenzymes. That is the big picture. So now our challenge is just to look at a few more details, and particularly to deal with this idea of how electrons passing through this transport system in the membrane can pump these hydrogen ions out here and create our potential energy. Okay? That's kind of where we need to go. And along the way, we'll do some accounting and see how many ATPs we get here and here and here. So you kind of understand, again, the big picture. Oh, by the way, if you have test corrections, remember they're due today. Okay. So let's do one group of test corrections is due today. If you had to do them through the mentor, they're not due till next week. So let's start out with glycolysis, okay? This process that occurs in the cytoplasm of the cell. This is not a one-step reaction. This is a multi-step metabolic pathway. Glycolysis begins with one glucose molecule and ends up by producing two molecules of this substance called pyruvate. So in glycolysis, we go from one glucose to two pyruvate. One of the things we're going to kind of keep track of as we go through these rearrangements of glucose is numbers of carbons. Okay? Glucose has 6 carbons, right? Remember C6H12O6? Okay? Pyruvate is a 3-carbon compound. So what does glycolysis accomplish? Basically, it chops glucose in half and makes 2 molecules. Along the way, some ATP is going to be produced by that substrate level phosphorylation. Glucose has more hydrogen than pyruvate does. So glucose is going to become oxidized. Where do those hydrogens go? They're going to be picked up by that partner, that coenzyme NAD, and in the process, NAD is going to become reduced. There's one other kind of major thing that happens in this particular process, and that is this process needs a little bit of input of energy to get going. And so this particular process of glycolysis both produces ATP, but it also uses some. What do you think? It's going to produce more ATP than it uses, or vice versa? Hopefully it's going to produce more ATP than it uses up, right? So there will be a net yield of ATP production. Does glycolysis release carbon dioxide molecules? We will answer that by looking at that equation up there. Is any CO2 lost? Nope. We have not lost any carbon, right? We started out with 6 carbons. We still end up with 2 times 3, 6 carbons. So there is no CO2 produced during the reactions of glycolysis. Okay, so this just kind of summarizes with a little bit more detail what we just talked about. And it breaks this down into some kind of categories of reactions. There are reactions that require an investment of energy, and then there are reactions that give you a payoff of energy. So again, what's it start with? It starts with a molecule of glucose. What does it end up with? It ends up with 2 molecules of pyruvate and some water that's produced as well. Okay? To get this started, for every glucose that goes through glycolysis, we have to initially put in 2 ATP molecules. This sometimes is referred to as priming the pump. And that's an analogy that probably doesn't make sense to you anymore. Have you ever seen those old handheld water pumps where you have to push this little handle lever and eventually water gets pulled up because you're creating vacuum and the pump starts running? Well, those pumps work because they have a seal in them that creates a vacuum as you operate the pump. And the seal used to be made out of things like rawhide. And rawhide would dry out as if the pump wasn't used frequently. And then if you don't have a good seal, you don't get a vacuum and you can't pump water up. So what people would have to do before they use the pump is to pour a little water down the pump to rehydrate that seal. And then when you start pumping the handle, you create a vacuum and it pulls water up. So you got to pour a little water in to get a lot of water out. Okay? Well, the cell has to do something similar. It has to invest some energy initially in the form of ATP to get the process going. Why? Because some of those early reactions in glycolysis are endergonic. The free energy of the reactants is less than the free energy of the products. So it's an uphill reaction. You've got to push it uphill using an investment of ATP. Later, there are some energy payoffs, and 4 ATP molecules per glucose are produced by substrate level phosphorylation. So what is our net total? 2 used, 4 produced. We get a net total of 2 ATP per glucose produced during these reactions of glycolysis. The other thing that's going to be significant potentially is that also there are some oxidation reactions occurring. Hydrogens are being stripped off of glucose, and they are being transferred temporarily to oxidized coenzymes, NAD in this case. So during glycolysis per glucose, 2 oxidized NAD become reduced. Okay? This represents, under the proper conditions, potential energy. Because remember I said when you add hydrogen to something, it increases its free energy? Okay? But at this point, there's no direct energy payoff from producing this compound. And actually, producing this compound creates a problem. What's the problem it creates? What if we just kept doing this? Could this keep on going on indefinitely? No. Why not? Because we would run out of the oxidized forms of NAD. And remember those enzymes that strip hydrogens off of molecules? What are they called? Remember what the prefix without means, without hydrogen? Dehydrogenase enzymes. Those dehydrogenase enzymes require this oxidized coenzyme. And if we have used this all up during glycolysis, then these reactions are going to stop. And what's really kind of tragic for metabolism is that the dehydrogenase enzymes work in this energy payoff phase. So the cell could still do this. It could still partially convert glucose into other things at the expense of energy. So we'd actually be using energy, but then we wouldn't get the energy payoff. So that's not good. Okay? So overall, the reaction is 1 glucose to 2 pyruvate plus 2 waters, 4 ATP formed by substrate level phosphorylation, 2 ATP used, NAD becomes reduced, and so we start running out of this cellular supply of oxidized NAD. So a cell cannot just do glycolysis alone. It would quickly run out of this, and everything down here would come to a screeching halt. So cellular metabolism contains glycolysis as part of the overall sequence, but it's got to be more than that. Okay? What specifically has to be done following glycolysis? Somehow this has got to be recycled back to this again. Does that make sense? Because that's critical to understanding pretty much everything we're going to talk about. Okay, so let's just show you a couple of the reactions and what they occur. And this is level of detail that I'm not going to be holding you responsible for. So you don't have to know the names or the structures of these particular compounds. You don't even really have to know at what points ATP is produced, NAD is reduced, etc. But I just want you to see it so you can appreciate a little bit about how glucose is ultimately rearranged, chopped in half, hydrogens removed, and eventually 2 pyruvates are formed. Okay? So in the first step of glycolysis, glucose is converted into glucose 6-phosphate. It's phosphorylated. Okay, you see the phosphate up here? And that happens at the expense of an ATP. This is the name of the enzyme that catalyzes that particular reaction. Don't have to know that name. Hexokinase. Glucose is basically rearranged a little bit. There's an enzyme called an isomerase, which moves things around a little bit. And glucose becomes converted into fructose, which is another different monosaccharide. This is fruit sugar. So this is fructose 6-phosphate. Fructose 6-phosphate is then phosphorylated again. So another ATP is consumed. So now we have 2 phosphates on this molecule. This product is called fructose 1,6-bisphosphate. This molecule is converted by an enzyme called aldolase into two products, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These two can convert into one another. Okay, so this can be converted into this can be converted into this. Glyceraldehyde 3-phosphate, though, is going to be acted upon by the next enzyme in glycolysis. So this is going to be pulled out into the next step. As the concentration of this decreases, more of this will be converted into this. Okay? So effectively what's happening is one molecule of this is being split into two molecules of this. That's the net result. This has got 6 carbons. Each of these have 3 carbons. Glyceraldehyde 3-phosphate is converted into 1,3-bisphosphate glycerate. That's an oxidation reaction. Okay? Now, I wish they had done something different on this slide. And if you've got your PowerPoint slides, you might want to do this on the slides. There really are, per glucose, 2 molecules of this being produced. There are 2 molecules of this being produced. There are 2 molecules of this being produced. And that's going to continue until we get all the way down to pyruvate. So they really should have a 2 by this to indicate that there are 2 of these produced per glucose. Okay? The number of ATPs and reduced NADs produced are per glucose molecule. So when we do our accounting, we always account for energy production based upon what you get from a glucose molecule. Okay? So in this particular step, one of the phosphates is pulled off. There's an enzyme that grabs this phosphate from this molecule and transfers it to ADP to make ATP. What kind of phosphorylation is that? That is substrate level phosphorylation. Okay? This molecule is rearranged a little bit into this one. A water molecule is pulled out to form this compound called phosphoenolpyruvate. And then the remaining phosphate group is pulled off to produce pyruvate. So when you look at pyruvate compared to glucose, there are less hydrogens present on that pyruvate than there were initially on the glucose molecule. Where did they go? They went here. And that's fine because this represents potential energy, but this is being formed at the expense of this critical coenzyme, oxidized NAD. And then we're producing a little bit of ATP by substrate level phosphorylation. Do you remember the number I told you last time in terms of maximum amount of ATP you could get per glucose? 38. Okay, remember that number 38. That's kind of the maximum. Gold standard for ATP production. So from glycolysis, we only get 4 out of 38. So it's a pretty tiny percentage. Okay? Around 10%, okay, of the ATPs produced by substrate level phosphorylation. We're accumulating this, but accumulating this is really a problem. Okay? So, once you have pyruvate, it's decision time. What happens to that pyruvate? Okay? The branch in the road is dependent upon an environmental factor. Is oxygen present or is oxygen absent? If oxygen is present, pyruvate goes through some additional reactions that make up a process called fermentation. If oxygen's present, pyruvate enters the mitochondria and goes through a different set of reactions called the citric acid cycle. So this would be the pathway for aerobic respiration. This would be the pathway for fermentation. Both pathways, aerobic and respiration, share the reactions of glycolysis. In fermentation, something different happens to pyruvate than it does in aerobic respiration. Let's take a look at fermentation first, because that's the one you're most familiar with, because you measured fermentation rates last week in lab. Pyruvate has had some of its hydrogen removed, but there's still hydrogen on that molecule. That's potential energy. In fermentation, that additional potential energy is pretty much wasted. The cell doesn't use it to gain additional energy. So what's happening from here to here? One big thing is happening, and that is during those reactions, the NAD is becoming oxidized again. So the hydrogen is being pulled off of the reduced NAD, and that then regenerates the oxidized NAD, which then can be used back here in glycolysis again. So fermentation is a little kind of self-sustaining system. Okay? These final steps from pyruvate to the end products of fermentation are occurring for this reason. Now, this represents potential energy. The cell does not trap any of that potential energy as NADH is being reoxidized again. So fermentation doesn't give you any additional payoff of energy production from glucose. So glycolysis produces 2 net ATP for glucose. Fermentation produces the same amount. Now, are all fermentative end products the same? No. Okay? There are a whole bunch of different types of fermentation. They all begin with glucose, but some end up by producing ethanol, some end up producing lactate, some produce butyric acid, some produce acetic acid. There are all kinds of different types of fermentative end products. And bacteria can produce a whole bunch of them. Okay? Anybody know an organism whose fermentative end product is primarily ethanol? Yeast, okay. And different strains of yeast produce different amounts of ethanol. Baker's yeast doesn't produce as much as brewer's yeast does. How about lactate or lactic acid? You may know any cells that produce lactate or lactic acid during fermentation? Muscle cells, okay? And that accumulation of lactic acid can contribute to muscle soreness, okay? Why are your muscle cells fermenting? Because if you go into vigorous exercise mode, you can incur something called an oxygen debt. And basically your circulatory system can't supply oxygen to your blood cells fast enough. So the cells become oxygen depleted, therefore pyruvate can't go into the mitochondria and go through aerobic cellular respiration. So it's basically forced to go through this fermentative pathway. So it's got some way to do this, to keep this going, so we can get at least our 2 measly ATP per glucose out of glycolysis. Okay? Can your muscle cells do this? Can they ferment and produce ethanol? No, it'd be kind of interesting, wouldn't it? Go out for a long run, incur oxygen debt, and produce ethanol. Why can yeast do this, but we can't? These are a set of chemical reactions. What's every chemical reaction controlled by? An enzyme, right? And what are enzymes? Most of them are proteins. And how does the cell know how to be able to make a particular protein? It's got to have the right gene, right? Okay. So we do not have the genes in our DNA for the enzymes necessary for ethanol fermentation. We do have the genes for the enzymes that allow lactic acid fermentation. Okay, so here's what happens during alcoholic fermentation, okay? We've got glucose being converted into pyruvate during the process of glycolysis. We get our 2 ATP produced by substrate level phosphorylation, and we get 2 NADs being reduced into this form. So we're depleting the oxidized NAD. And if this runs out, glycolysis stops. Therefore, we don't get this. Okay? So what happens in fermentation? There's a couple of additional steps. Pyruvate, which can't go into the mitochondria because oxygen's not present, is enzymatically converted into a compound called acetaldehyde. Pyruvate has 3 carbons. Acetaldehyde has 2 carbons. So where's the carbon go? It's lost in the form of carbon dioxide. Enzymes that remove carbon from substrates and produce CO2 catalyze a process called decarboxylation. So these particular enzymes are decarboxylases. Okay? So this enzyme that catalyzes this particular reaction is going to be a decarboxylase. So again, per glucose, we produce 2 molecules of CO2. Those were those bubbles that you were measuring in the fermentation process. The bubbles were displacing the liquid, and the level of the liquid was falling in those centrifuge tubes, and you were measuring those volume changes. Okay? But we still haven't solved our NAD problem yet, have we? All we've done is we've taken a carbon off of this. In the final step of fermentation, acetaldehyde becomes reduced into ethanol. This has more hydrogen than this does. Okay, a hydrogen is added right here. Where does it come from? It comes from the reduced coenzyme, and as this gives up its hydrogen, it becomes reoxidized again. Okay, so do you see how fermentation is a self-sustaining process? Now, is this much energy? No. Can yeast cells survive on that 2 ATP per glucose? Yeah, they can. Okay, they have pretty simple demands. But can your brain cells? No. That 2 ATP per glucose is not enough energy to maintain the highly organized brain cell. And so brain cells start dying, even though there may be a desperate attempt for them to stay alive by going through the process of fermentation. But they don't do ethanolic fermentation. They don't have the enzymes to do that. So, what can muscle cells and other human cells do? Lactic acid fermentation. Glucose, converted to pyruvate, still got our 2 ATP, still got our 2 reduced NAD. So we've got the NAD problem now, the reduced NAD problem. Pyruvate is then converted into lactate. This has more hydrogen than this does. Where does it come from? Here. Is there any decarboxylation associated with lactic acid fermentation? 3 carbons here, 3 carbons here. So lactic acid fermentation does not produce gas. And that's probably pretty good. Because it's one thing for your muscle cells to accumulate this lactic acid, which can make them sore, but you want your muscle cells producing a lot of CO2 gas? Probably not. Okay, you want your muscle cells swell up as gas is produced during fermentation. Does fermentation make sense? Okay. This is the key that explains why we need that add-on to glycolysis to keep glycolysis running. Okay. So what if oxygen is present? Well, if oxygen's present, then pyruvate has a different fate. We've got pyruvate out here in the cytoplasm, and pyruvate is going to move from the cytoplasm into the mitochondria. It's going to be transported. And it's going to have to cross that outer membrane of the mitochondria. It's going to have to move through this inner membrane. And eventually pyruvate ends up here in this matrix of the mitochondria. And when it gets there, it encounters a whole bunch of different enzymes which are going to then further process the pyruvate. Okay? What's that further processing involved? Well, pyruvate's still got some hydrogen on it, doesn't it? And if we pull that hydrogen off of it, we can potentially extract energy from pyruvate. And ultimately, if we pull all of the hydrogen off of pyruvate, what are we left with? A molecule with carbon and oxygen, carbon dioxide. Pyruvate, when it enters the mitochondria, is initially processed into a molecule called acetyl-CoA. And then it's acetyl-CoA that literally feeds into this set of reactions called the citric acid cycle. Technically, this step right here, of pyruvate being converted into acetyl-CoA, is referred to as the transition reaction. Okay? When we start doing a ledger of ATP production, what I'm going to do is I'm going to lump these two things together. And I'm going to lump together this transition reaction along with the citric acid cycle. So be aware of that. Okay? So let's take a look at kind of what's happening overall, big picture. Remember, pyruvate has 3 carbons on it. And it's still got some hydrogen on it too. In this transition reaction, several things happen. Some hydrogen is removed from pyruvate. Where does it go? It goes to the oxidized NAD, reducing it. Okay? A coenzyme is temporarily added to the processed pyruvate molecule. This coenzyme is a coenzyme called coenzyme A. And one of the carbons is split off of the pyruvate. So this is a pretty interesting enzyme that catalyzes this reaction. This is a complicated enzyme, and it's referred to or named the pyruvate dehydrogenase complex. Okay, well, you should know what this means, dehydrogenase. It's pulling some hydrogen off the pyruvate. In reality, this enzyme is also a decarboxylase because it's removing some CO2. And it's also able to grab this coenzyme A and actually transfer it to this molecule. So the end product of this pyruvate dehydrogenase complex is a molecule called acetyl-CoA. So we've gone from 3 carbons now to 2 carbons. Where's the carbon? Right here. It's been liberated in the form of CO2. Acetyl-CoA still has some hydrogens attached to it. So it enters this set of reactions called the citric acid cycle. Now, here's something important to know about cycles. Cycles, if they begin here, always have to get back to the same starting point again. So if this cycle is going to continue, whatever is fed into it is basically going to have to come out at some point in time. So what do we feed into this? We feed in 2 carbons at a time into the cycle. If this cycle is going to be continually turning, we got to get rid of what we fed into it. So eventually 2 more carbon dioxide molecules are going to be produced as a result of this citric acid cycle. So what's been accomplished by this transition reaction plus citric acid cycle? The 3 carbons in pyruvate have been converted into 1, 2, 3 CO2s. It's been completely oxidized. Okay? Where did those hydrogens go? Some are picked up by NAD. Some more are picked up by NAD here. And also now we've got this new coenzyme FAD for the first time. That picks up some hydrogens. We also are able, during this process, if we have some inorganic phosphate present, to actually make ATP by substrate level phosphorylation. Okay, on this slide, these numbers represent what's produced per pyruvate. See you later. Okay, this is per pyruvate. How many pyruvates do you produce per glucose? 2, right? So if we're doing an ATP accounting, we have to multiply everything on this slide by 2. Okay? So 2 ATP per glucose are produced by the citric acid cycle. They're produced by substrate level phosphorylation. But what else is being produced? 2 reduced NAD here per glucose. 6 reduced NAD here. 2 reduced FAD here. Well, that's a potential opportunity because these are potential energy. Okay? They've got hydrogens. And we also got the hydrogens hanging around on the reduced coenzymes that were produced during glycolysis, right? So there's an opportunity there. But it also is a problem. What's the problem? We're running out of this. And we're running out of this. And if we run out of these oxidized coenzymes, this whole process stops. So then what would pyruvate be forced to do if this stops? Go through fermentation. Okay? At least in fermentation, you can recycle your coenzymes. So these reactions basically finish the oxidation of pyruvate. Pyruvate has been partially oxidized during glycolysis. This finishes the process and removes the remaining hydrogens. So we accumulate a lot of these reduced coenzymes, and we make a little bit of ATP. And what do we produce as a byproduct? CO2. So the CO2 starts to build up in the matrix of the mitochondria. So what happens when it gets in a really high concentration here? It moves across this membrane. It moves back out into the cytoplasm. When it builds up into a high concentration in the cytoplasm, then as blood supply comes to that cell, the carbon dioxide is transferred back to the blood. The red blood cells and the plasma, particularly of the blood, pick up that carbon dioxide and transport it back to the lungs. And then it's exhaled. Okay? So when you eat sugar, you eventually breathe out carbon dioxide. You've produced some energy, but how much energy have we produced so far total from a glucose molecule? How many during glycolysis? 2 net, right? Produce 4, use 2 net. How many here? 2 more. So we're up to 4, but that's not 38, right? And we have this NAD-FAD problem. So in aerobic cellular respiration, there's one more thing that has to happen. These have to be recycled back to these. And how do they do that? That happens during the 3rd phase of cellular respiration, the electron transport chain. And as that recycling occurs, which is happening in this inner membrane of the mitochondria, as electrons are being transferred from one compound to another, eventually going where? Where do they end up? What finally grabs the electrons in aerobic cellular respiration? Oxygen, right? Make water. We pump hydrogen ions out here, and that creates the potential energy to then allow ATP to be produced. And during that process of oxidative phosphorylation, we get the additional 34 ATP per glucose. So now what we got to do really is next time come to grips with how does this electron transport chain, as it's recycling coenzymes, pump hydrogen ions? How does it charge that battery, if you like, that the cell then uses to make ATP? So what we'll do next time is, just to reinforce this a little bit, is we'll run through some of the individual steps of citric acid cycle, just like we did glycolysis, so you can see what's happening, okay? But the important thing are these big picture items. That's really where I want you to concentrate. Okay? Have a good weekend. We'll do this again on Monday.
Dr. Browder.
Yes.
Good, this one, I had the lenses on. I guess I just like people have that know how to.
Oh, you did?
Yeah,
Okay.
use it.
Let me look back at this again and see if I need to adjust that a little bit.
All right.
Okay? Would you make a quick note on there to check that question?